So in our previous unit, we've already talked about different types of genetics and different types of inheritance. How do you inherit a specific trait? How does a parent pass along this trait to their child? But now in this unit, we're going to continue talking about genetics, but we're going to talk about how do scientists or doctors study those genetics. And so in Unit 3.4, we're really, first of all, going to be looking at different ways we can see how a parent will pass on the trait such as hemophilia or cystic fibrosis to their child. So we're going to be on, actually it's going to be notes 3.4, not 4.1, and we're going to be on page 65. So again, this is in Unit 3.4a. All right. So when we're studying human genetics, there are a few problems or a few little setbacks for us. First of all, we can't study human genetics the same way that we would study bacteria. There are genes in bacteria that can be studied. There are genes in mice that can be studied. But we can't look at them the same way. The reason for that is because it is going to be irresponsible to perform a test cross on humans. Now, what is a test cross? An example here would be if we had two different types of snakes. We had one snake that is colored and another one that is albino. It doesn't have any color. It's all white. Okay, it lacks those pigments. But the question is that we know that if our snake is albino, doesn't have any color, then it received two recessive alleles, two little A's. But our snake that has color to it, well, we know that it has one dominant allele, one big A, but we don't know for sure what the second allele is. We don't know if it is another big A or a lowercase a. We don't know if it's homozygous dominant or if it's heterozygous. So in a case of animals, well, it's really easy to figure that out because what we can do is we can do a test cross or actually literally mate them together. And by looking at the results of their offspring, we could actually see, or we can actually determine, what was this snake's genotype. If the snake ends up having babies or offspring that all are colored, so all have a big A, lowercase a, then we know that this first parent snake, was big A, big A. But if we end up having snakes that are half colored and half albino, then we know that this parent snake was heterozygous big A, little a. All right? But for humans, if we don't know what the genotype of a parent is, we can't just do a test cross. We can't just tell them to go have offspring so we can figure this out. So the test cross method is going to be to determine what type of genotype a dominant phenotype has. So again, let's say a parent has Huntington's disease. We don't know if they're homozygous, have two big letters, or heterozygous, a big letter and a lowercase letter. So in order to figure this out, we could do a test cross, but just not in humans. We're not allowed to do that. The other problem when studying human genetics is that humans have a really long lifespan compared to other organisms. And in order to study several generations, it would take a really, really long time. And the other problem is that we have a small number of offspring. Usually, how many kids do a set of parents have every time? Usually just one child. And we also have a long gestational period, which means a long pregnancy. Other organisms think of a puppy. They might be pregnant for about 3 or 4 months, and at the end of the 3 or 4 months have 3 or 4 puppies. Okay? It's a very short lifespan. Dogs usually live maybe 15 years is a really high number. They have lots of offspring, or they can have lots of offspring within those 15 years. They might have 10, 15 puppies if they give birth 3 or 4 times. And they have very short gestational periods. So something like a dog would be much easier to study than a human. So we do have some techniques to study human genetics. One of the first is doing a population sampling. And a population sampling, the best way to understand that is to look at this picture. And you actually want to redraw this picture right beside this little section of notes. And it's really simple. The first thing it says is you want to draw a big circle, and inside the big circle you want to write population. Then you want a small circle underneath that that says sample. In a population sampling, what you do is you take a small sample from the main population, maybe all of Charlotte, take a small sample. Once you get your small sample, you can take the data from your small sample, and then you can create an inference on what would apply to the rest of the population. For example, in all of Charlotte, how many people are hemophilic? How many people have the blood clotting disorder? Well, let's say we take 100 people out of the population, randomly select them. Once we have our random 100 people, well, how many out of 100? Maybe we only have 1 out of 100 people. Well, then we can infer that if there are 300 people in this population, then 1 out of each 100 persons would have the disorder. So maybe 3 people in the whole population have it. But do make sure you redraw this picture. You have your population, your sampling, your actual sample, and your inference. Okay? You're going to collect data from the sample and apply it to the entire population. Another way to study human genetics is by looking at pedigrees. And if you think about this, a pedigree, another word for this is just a family tree. A lot of times when you go to the doctor, they'll ask you, does your family have a history of heart disease? Does your family have a history of diabetes? And those are all things that we can kind of graph or put into a family tree. We call it a pedigree in biology. And then to see if there was a trend, if it's been passed on from certain people. So it's a graphical record of inheritance of a single trait over several generations. This is the good thing about a pedigree. Then you can look at it over several generations. Okay? So today we're going to go a little more into pedigrees, looking at how do we read pedigrees, how can we use them to track genetics, track genes. And the first thing that we need to know is that a shape is going to represent individuals in a pedigree. And then the connecting lines will represent relationships. So you do have a little key in your notes here. A circle represents female, square represents a male. If you're not colored in, then you're not affected, unaffected, or you can also say don't have the trait, okay, or no trait. And if you're affected, then you have the trait or have the disease, okay? So if you look at this picture really quick, just to talk about it, this square that's colored in, what does this mean? If we're talking about a disease like PKU, does this person have the disease? Is this person a male or female? If you look over here, this person is colored in half. You don't always see this in a pedigree. Sometimes they don't color in half at all. But is this person a male or female? Does this mean that the person is affected, has the disease, or is unaffected, doesn't have the disease? Well, for right here, the first person, this is going to be a male. Does he have PKU? Yes, he has PKU because he's colored in. For this person, male or female? Well, this is going to be a female. And they're only colored in halfway. So what could that be describing? PKU is a recessive disorder represented by a little a. So this man, what is his genotype? Little a, little a. Well, what does it mean about this female? This means that the female is actually heterozygous, big A, little a. And what does that mean about her and PKU? This actually means that she's a carrier. So underneath your key box or above it, you'll want to add in a circle that's half colored in and a square that's half colored in and label this as a carrier. But keep in mind that they don't always label carriers in a pedigree. Sometimes you have to figure it out on your own. So in pedigrees, there are a couple of questions that you need to consider. And these are going to help you to determine, is this a dominant or recessive trait that's being tracked on the pedigree? And if it's dominant or recessive, or actually if it's a recessive disorder, is it autosomal or is it sex-linked? What you need to know about question number 2 is that if it's an autosomal genetic disorder, that just means that it's not on your X chromosome. So you'll want to add in here that sex-linked means on the X chromosome. And I'm going to shorthand that CHRMS, chromosome. Make sure you take a second to copy down all of these answers to these questions. Are there any children who have the trait but both parents don't? If this is the case, then this means that the trait is recessive. If not, then a parent with the trait definitely passes the trait to the child. This would be recessive. This second portion here would be dominant. If the disease ends up being recessive, a child can get the disease from both parents that don't have the disease. Then you have to determine, is it autosomal on your regular chromosomes, or is it sex-linked on your X chromosome? Well, if the daughter has the disease, did her dad have her dad? If her dad has the disease, then yes, it's probably sex-linked. If the mom has the disease, does every son have the trait? If this is yes, then it's also sex-linked. Now, if it is sex-linked, can a son inherit from the dad? The answer to that is no, because the son only receives a Y chromosome from dad. And remember, if it's a sex-linked disorder, you get that on your X chromosome. So make sure you pause and copy the rest down if you need to. Later on, we're going to look at each of these pedigrees and look through some of these examples here, those questions that you just looked at. So we'll look at those later on in class. Here you want to copy down 2 more things. The first is about the Human Genome Project. You're going to want to pause, read through this, and copy down the rest of the notes on the Human Genome Project. How did scientists figure out what all of our genes or what all of our chromosomes coded for? And the last thing that you'll want to copy down for today's notes is some prevention and treatment possibilities for people with genetic disorders. We can go through genetic counseling, and we can also have some treatments. They don't cure the disease, but they can make the person much more comfortable. So make sure you also pause here and copy the rest of these notes. And the final thing for today is your summary box. In your summary box, I would like you to write down 2 sentences about 2 different facts that are most important in this unit. Okay. Once you finish that, you'll be done for today, but make sure that you do write down 2 summary sentences of this section of notes.